Good afternoon, students. Today in this lecture class, I'm dealing bioprocess related with the bioreactors. Okay, this comes under the paper Bioprocess and Environmental Biotechnology, that is Unit 2, Bioreactors and Downstream Processing. Okay, today I'm dealing bioreactors, that is significance of impeller, baffles, and sparger. To understand all these six, first we need to understand what is this bioprocess, and then we need to understand about the stir tank fermenter and about the different parameters of this particular bioreactor. Okay, so bioreactor or fermenter almost one the same, slight variation can be observed here. And in this bioreactor, you need to understand about the design and about the functions. Usually we use the terminology like bioprocess. So what is this bioprocess? Bioprocess in industrial biotechnology refers to the use of living cells. Living cells in the sense like microorganisms and their components like enzymes to manufacture industrially important products or to carry out a specific chemical transformation on a larger scale. We are using this particular bioprocess. This field also integrates biology and engineering principles to develop to optimize processes for the production of wide array of goods. Wide array of goods means that is nothing but the variety of microbial products. Okay, that some of the core concepts like a biocatalyst, bioreactor, then becomes also we study here upstream processing, downstream processing, fermentation, media. So all these we are studying to obtain the different varieties of the microbial products like pharmaceuticals, food and beverages, and biofuels, different agriculture product, and genetically modified organisms product, and so on, so forth. Okay. Now let us focus on this bioreactor design and their functions. All right. The process of fermentation is usually done to maintain the growth of the industrial microorganisms in an environment which usually stimulates the synthesis of the desired product that are commercially used, right? So we are in this particular fermentation process or in the bioreactor, we are focusing more on industrially important microorganisms which could give a good yield, a novel product. Those organisms we are going to culture as subject in the fermenter vessel. The fermenter is a large vessel in which organisms are maintained at a necessary heat, a required pH, then the air we need to provide if that is if that is aerobic microorganisms, we need to provide the proper optimal air and also the substrate concentration. So in the fermenter vessel, we need to provide all these requirements for the growth of the microorganisms in an optimal condition, right? Then fermentation systems must be efficiently controlled in order to optimize productivity. This optimal content is very, very important for the to obtain a good yield or maybe good product and product yield, and also to ensure reproducibility. So all this matters a lot in the fermenter vessel while designing and also while during the process. Fermenter or bioreactor refers to a device that provides all the basic necessities important for biological product extraction. Okay, a fermenter contains different devices that help to maintain the environmental factor inside, which in turn leads to the production of the biological products. Therefore, what is the main objective of the fermenter? The main objective of the fermenter is to maintain a controlled environment that supports the growth of the bacteria or any other organisms. What are those controlled environment? That may be aeration or agitation, pH control, temperature regulation, or antifoaming agents, so on, so forth. So all this one has to keep in their mind, and then they should work on this particular fermenter vessel. The key factors in the fermentation process control are, that is aeration, the device which is used for the aeration, we say that as the sparger. Then for the agitation, the device we used here, that is the impeller. Then temperature regulation, different inlet, different cooling jackets will be there. Okay, to reduce the temperature, we use cold water that will be sent to the pipe, and to increase the temperature, we can use the heating coils so that we can increase the temperature. Okay. And then foam control is also very important, as this is the in most of the aerobic fermenter we are using the liquid media. During the agitation, or aeration, there will be the formation of the foam will be there. Definitely, the foam must be controlled by using anti-foaming agents. Then pH regulation. So this is also important. So all these parameters matters a lot during the fermentation process. What are those? Aeration, agitation, temperature regulation, foam control by using the anti-foaming agent, then pH regulations. Apart from all this, the most important one is also baffles also will be adjusted to the fermenter vessel to prevent the swirling or maybe a vortexing. Otherwise, if there is no baffles, that will hinder the productivity formation, also breaking of the cells possibility can also be observed. So all these different key factors matters a lot during the fermentation process. All that should be controlled. What are those? Aeration, agitation, temperature regulation, foam control, pH regulation, and about the baffles. Okay, so this is one of the diagrammatic representation, the typical stirred tank fermenter. Also, we can say this as aerobic fermenter. Okay, we can see the internal view of the aerobic fermentation. Okay, so these are the impellers. You have in your syllabus about the impeller, then about the sparger, and then about the baffles. You need to focus on these three key factors. What are those? Impeller, sparger, and baffles. Apart from these three key factors, you need to focus on other important key factors of the fermenter or vessel. Okay, so we say this as stir tank fermenter, also a typical fermenter. Why we say this as stir tank? Because there is an impeller is there for the mixing of the media and the microorganisms in a uniform condition. So that for that reason, they call this a stir tank for the stirring purpose. No, that's why they call this as the stir tank fermenter. So what all you can see, these impellers are stir tank fermenter, which is run by using this particular. You can see here, this is the motor which is connected to the shaft. Then you can see here, this is the steam valve. Okay, this is the sterile seal, and this is the viewing port will be there. And these are the impellers. Okay, you can see here, this is the media and the microorganisms. And this is the cooling jacket, cooling water inlet pipe. And here you can see this is the steam valve. And this is the harvest outlet valve. This is the sterile air inlet where the sterile air will be sent, and that the air will be appearing from this particular sparger. You can see here the bubbles here. This is the sparger with the high pressure air. Bubbles, gas bubbles will be sent here to this particular viscous medium. And there is a culture broth inside. And this is the baffles to prevent the vortexing. And this is the cooling water out. And this is the filter, and this is the exhaust pipe. And you can see here to maintain the pH, acid-base reservoir and the pump. Okay, pH controller can be observed. So all these are the different parameters one has to keep in their mind during the stir tank fermenter. Okay, next, this is the diagram written by me. So the same diagram. Okay, so same thing here, motor is there, culture nutrient addition, sample line, temperature sensor control unit, cooling water inlet in, and these are the impellers, and the interior baffles will be there. And this is the sparger. Okay, and also biosensor unit also can be observed here. So this is all about your stir tank fermenter or maybe a typical bioreactor. You can see here, this is in some of the research laboratory. You can see this particular steel fermenter vessel. In contrast to this steel fermenter vessel, we'll also get the glass one, a tabletop fermenter vessel, which is made up of borosilicate, which can withstand the high temperature and also the pressure. And this is the head plate, which is totally sealed. And you can see here many different inlet pipes can also be observed here. Okay, for this is the huge fermenter vessel. Very good to see here. These are the baffles which are present in the wall of the bioreactor. And here you can make out these are the blades, which we call this are the impellers for the mixing of the media. Okay, large bio industries will be there. No, there usually we find this such type of big device, that is the impellers. Now, let us understand one by one about this fermenter vessel and the parameters. The fermenter vessel, which is usually consists of a large vessel to culture the microorganisms and also for the product formation. The body of the vessel, which is made up of thick stainless steel sheet or thick glass, which is made up of borosilicate, which can withstand high pressure, okay, and also temperature and also low pressure. temperature up to 180 degrees Celsius. And usually these fermenter vessel is a closed cylinder connected with various pipes, letting for in culture medium, inoculum, then acids or alkalis for the pH control and also antifoam agents and etc. Okay, I know, but you know better. This is inoculum, mean that is nothing but the microorganism. Okay. Outlet pipes for periodical sample collection can also will be there at the bottom of the fermenter vessel. Harvest lines for the periodic collection of culture broth and product recovery will be done through the harvest lines. And other several online control system for monitoring temperature, pH, cell density, then oxygen concentration, carbon dioxide level indicators, a nutrient content indicator that will be there. As I use the word here, online means most of the nowadays fermenter vessels, which are automated systems, okay, based on the automated system, whatever they require, what culture medium or acid base to be adjusted or cooling effect, if they need or heating effect they need, okay, if they need any kind of, that everything that will be automated one that monitored by the system itself. Okay, all the inlets and outlets are completely sealed with the membrane seal to prevent the entry and exit of any kind of liquid. What are the inlet pipes and outlet pipes will be there? Those pipes compulsory that must be sealed, otherwise contamination chances can be observed. For example, what are the inlet and outlet pipes will be there? That will be always sealed by the butyl nitrile rubber seals or butyl rubber seals, butyl airtight why we are using butyl rubber seals? Because this butyl which will makes the airtight and gas impermeable will be done. That's why we are using this particular inlet and outlet pipes which are sealed totally by using the chemical called butyl. It means the pipe, all the inlet pipes and outlet pipes only compulsory that should be closed further to minimize the contamination. That is very much important. And also the whatever the vessel is there, that vessel should also withstand to the if it's stand to the temperature, and also the content, whatever there should be enough space for the formation of the product also. The vessel should be strong enough to hold large volumes of culture broth and pressure that could be generated due to the gas production within the fermenter vessel. The volume like a few thousand liters to more than 100,000 liters or maybe a gallon level, the bigger fermenter vessel can also be observed. And these vessels are thick gauge which are made up of stainless steel, okay, that can withstand to the temperature up to 180 degree or more. And the vessel should be completely free from any kind of leakages, otherwise long-term operations will be difficult, and due to the leakages, contamination chances will be there. That's why there should not be any kind of the leakages. The size and the internal volume of the fermenter are designed always with the knowledge of bioengineers and fluid dynamics specialist people, and also microbiologists who provide the what amount of the inoculum to be added or what amount of the culture broth to be added and what kind of product that is going to produce, what kind of what level pH we need to adjust, and temperature and aeration, everything that will be, it's like a collaborative work. Okay, another microbiologist is not doing all the work. Here microbiologists, biotechnologists, bioengineers and fluid dynamics specialist people, by taking the knowledge of all the people, this particular bioreactor or fermenter vessel will be designed. Because once the fermenter is that fermenter will be always designed in such a way that product for the product formation also there should be given enough space apart from the culture medium and apart from the allowing for any kind of inoculum. In the fermenter vessel, maintaining the sterility in the entire system is very, very important. Okay, aseptic condition and the fermenter vessel must be compulsory devoid of any kind of contamination. Monitor the rate of product formation and control quality of the product is very, very important. Smaller fermenters are also available, which is less than 10,000 liters, which is made up of thick borosilicate glass jars and can be used instead of steel vessels. Okay, 10,000 or maybe a tabletop bioreactors will be there that can be used in the laboratory. Now let us understand here the peripheral parts and accessories of the fermenter vessel, adequate reservoirs for the supply of nutrient medium, inoculum, acid, alkali, antifoam reagents, water needed for the cooling, etc. This must be provided along with the fermenter vessel. All the reservoirs very, very important. That should be filled. Adequate devices for aeration is very important. That is the sparger. Then agitation, impellers for fermenting broth. And further, it should be allowed for microbial metabolism at optimal level. And airflow rate should be controlled by using the rotometer. That is, the air should be sterilized before letting into the vessel. Rotometer is very, very important here. Then next, coming to the impellers. These impellers are the device used for the agitation of the liquid broth, and they are rotated through the motor connected to a shaft. And the sparger here, sparger means this is the a device, a structure that allow sterile air to enter into the vessel in the form of bubbles or gas bubbles, and these are meant for aeration purpose, right? Impellers for the agitation, spargers for the aeration. Further, to control the temperature generated during the fermentation, the vessel should be provided with an external cooling jacket through which water can be circulated continuously to remove heat. To remove heat means we are going to send the cool water through the cooling jacket. Sometimes what happens here, temperature may go down, okay, due to endothermic reactions, temperature may go down. In such cases, heating devices are to be provided, like heating coil will be fixed over there. Through heating coil, when you switch on, the optimal heat temperature will be generated over there. So the vessel should be attached to its inner wall, the vertical plates called baffles to prevent vortexing. So far, we went through about the impellers, then sparger, right? Now the next terminology, it is the baffles. We are using baffles in the fermenter vessel. So what is this baffles? It's a device which are present inner wall of the fermenter vessel, okay, to prevent vortexing. The vessel, the fermenter vessel or bioreactor should be attached to its inner wall, which is look like a vertical plates, okay, which is used for to prevent the vortexing or swirling. ಈ ಸುಳಿ ಅಂತ ಹೇಳ್ತೀವಲ್ಲ, ಆ ಸುಳಿ ಅಥವಾ swirling ಆಗೋದನ್ನ ಅವಾಯ್ಡ್ ಮಾಡೋಕೆ we are using this particular baffles. And these are provided with and further the inlet pipes to receive the culture medium, okay. Apart from baffles, apart from here temperature regulation, cooling jackets and the heating coil, okay, there also you can see some of the inlet pipes to receive the culture medium. If for example, during the culturing process or bioprocess, the nutrients exhausted already. So still microorganisms are there. The microns, if they need nutrients means, and there must be a inlet pipes to receive the culture medium, right? Culture medium is nothing but the nutrients, the nutrient utilized by the microorganisms. Even sometimes inoculum is very less, microorganisms are very less, means there must be a compulsory inlet pipe to allow the inoculum or microorganism. That's why the fermenter vessels which are provided with the inlet pipes to receive the culture medium, that is culture broth, and also the inoculum, okay. And also they have an adequate outlet pipes for harvest of the post-fermentation broth and product recovery. There should be number of online controls with automation to monitor various parameters like temperature, oxygen concentration, carbon dioxide concentration, pH, conductivity, cell concentration, nutrient salts, etc. I hope you're able to follow. If I'm going very quick and fast, kindly you can pause this video and make a note of this particular information. Okay, most of the industrial bioprocess, whatever we are getting like microbial products, which are obtained by using the aerobic microorganisms. Okay, so here the most of the industrial bioprocess are aerobic and are carried out in the liquid media. Whenever we see the liquid media, there compulsory you can see the impellers. Impellers will be there, means there will be compulsory, there will be a spargers will be there for the aeration. So fermenters have to be provided with stirring devices, that is the impellers, and are called stirring tanks. Due to the presence of stirring devices, we say that particular typical fermenter as stirred tank fermenter. Okay, stirring is the mechanism to provide the continuous aeration. If there is no stirring, means what happened? The air will be at the bottom only. The air cannot be moved to that particular upper top level of the culture medium. So the impellers also will take the mix the uniform content, and also we take the air or gas bubbles to the at the top of the fermenter vessel also. Now let us focus on one by one aeration, agitation, and about the baffles. So aeration, it means that is to send the air inside the fermenter vessel by using the sparger, right? Most of the microbial fermentations are aerobic and need continuous supply of air, which is totally free from any kind of microbial cells or dust, and even the cells, any kind of other contaminants must not be there, right? So microbial fermentations of aerobic condition compulsory they need the air, and even anaerobic fermentations like alcoholic fermentation they require in the beginning they need the supply of air for the rapid growth and biomass formation, and later they switch on to the anaerobic conditions for the product formation. Okay, so for the aeration we are using the device called sparger. The sparger, which is responsible to provide the air bubbles or gas bubbles. In aerobic cultivation process, the purpose of the sparger is to supply adequate oxygen to the growing cells. So it is the purpose of the sparger. In the sparger, we are going to send the sterile air, right? Sterile air is passed into the fermenter vessel with the pressure so that air is properly dispersed in the viscous liquid medium. Okay, with the proper pressure, the air must be sent from the bottom to the top of the fermenter vessel in association with the impellers. They need a proper pressure also. Sterile air may be obtained by passing air through the filter containing a glass, wool, carbon particle, and other fine particulate substance which can trap microorganisms and particulate matter in the air. As we are sending sterile air only, if by chance any kind of contaminations mean this glass, wool, or maybe carbon particle or fine substances which is going to trap that particular organisms in the inlet pipe of the air. Beyond the filter air, beyond the filter, they filter the air passes through the sterile piping and connects at the bottom of the bioreactor vessel to a device called sparger. To promote aeration in the stirred tanks, the sparger is usually located directly below the agitator. Okay. So what I said here, to promote aeration in the stirred tanks, okay, the sparger is usually located directly below the agitator. Okay, below the agitator at the bottom of the fermenter vessel, you can see the presence of the this particular sparger. You can see here some of the sparger here. This is a sparger with the minute holes. Okay, and this is also sparger with the minute holes can be observed here. And these are some of the micro spargers. Okay, with the minute holes that will be used. And even there are a variety of the spargers you are going to get here. The small air holes can be observed here. This is the one of the sparger, large and fewer air holes facing up or down, and sparger ring can also be observed here. And these are the air bubbles which are generated from the sparger straight pipe with the holes. Okay, vertical pipes can also be observed as a sparger, or maybe a small minute holes on the disc can also be observed. Our vertical pipes can be observed. To that pipe outside there'll be a perforations will be. Perforation means holes can be observed. Vertical pipes also will be there. Fitted with a sparger. And also we can use to generate the air bubbles. We can also use the diffuser. Some of the diffuser can also be used. To this also, the sterile air can be sent. Okay. So sparger usually a horizontal circular plate with the minute holes. You'll get the circular horizontal plate cannot be observed. There minute holes can be observed. Or an erected pipe, like a pipe. Okay, erected pipe with the holes on the body of the sparger can also be observed. Circular one, on top of that erected one. Erected pipes, small erected pipes will be there. On that pipe, below perforations can be observed. The holes in sparger should be of the diameter ranging from 4 to 8 mm. That is again depends upon the what type of product you are getting from the fermenter vessel and what type of organisms used and what is the viscosity of that particular media. Everything matters. By going through that one can design the sparger. Bubbles releasing from the sparger are not useful if they are too big or too small. Adequate size should be maintained in the fermenter vessel. Smaller the bubble size, greater the surface area of the air that comes in contact with the cells. And injecting too small air bubbles will require greater pressure and it is very expensive. Usually we are playing in the tabletop fermenter. In the larger fermenter vessel, too small air bubbles are very difficult to operate. And small pores may easily get clogged due to the cell position. What are the holes will be there? If that is too small, means cells will only go and sit there and that will be clogged. It's very complicated if that is too small. Okay. And too big is also very complicated. Too big also not manageable. So that should be adequate. Aeration is very important. Fermenters meant for growing filamentous fungi require different kind of spargers with the holes of larger diameter, that is about 8 to 10 times the diameter specified for the organisms. Okay. I hope you're understanding about the sparger. Okay, during aerobic fermentations, molecular oxygen must be maintained at optimal level to ensure maximum productivity. Okay, so that is about the sparger, which is used for the aeration, okay, to send the gas bubbles to provide the aerobic respiration. Then now let us focus on agitation. For the agitation, we are using the device called impellers, which is brought about by stirring and mixing of the fermentation media. This brings about the following characteristics. Why we are using agitation or impellers? For the dispersion of the air in the liquid solution. Okay, for the proper dispersion of the air, whatever the sparges send air into the medium, that air must be properly dispersed throughout the culture medium. For that reason, we are using the agitator or impellers. What purpose? Dispersion of the air in the liquid solution. Apart from that, homogenization to equalize the temperature and concentration of the nutrition throughout the fermenter. Okay, for the homogenization of the content. Then dispersion of the immiscible liquids, we are using the agitator. Then these are used for mixing of the contents of the reactor, which keep the cells in the perfect homogeneous condition for better transport of nutrients and oxygen to the desired products. Okay, so because of all these reasons, we are using the baffle or maybe agitator, and process is known as the agitation. I hope you're able to understand here, right? Next, as you can see, these are the agitator, okay? This is the agitator shaft and these are the impellers. You can make out here, these are the impellers. Okay, this is the stainless steel from a bioreactor and this is the glass one, okay, bioreactor. And you can see here, this is a large fermenter vessel where you can see the blades like this, okay? Different design of blades can also be obtained in the different fermenter vessel. And these blades constructed mainly based upon the what kind of product to be obtained from the fermenter vessel. So you can see this is the baffles. Okay, this is the baffles which is present inner wall of the fermenter vessel. It's a huge bioreactor where you can see the man has went inside through the ladder and checking. Further, agitation is important for oxygen transfer in aerobic fermentation, right? The air bubbles released from the sparges are distributed uniformly in the culture vessel medium by the use of impellers. Okay, that is nothing but the agitators. Impellers are, what are impellers means? You can write this one. Okay, impellers are rotating devices with blades connected to a central shaft, which in turn connected to an electric motor that rotates at a specified speed depending upon the system. Okay, and these impellers play an important role in mixing up the inoculum, culture medium, and also transporting of oxygen bubbles to the entire container and also for the homogenization purpose. Okay, the impellers are of different design and sizes based on the requirement and also based on the productivity. The impeller diameter is about 1/3 of the vessel diameter. Okay, some commonly used impellers are like disc turbine, concave blade turbines, hydrofoil impellers, marine propeller. There are so many other impellers will be there. Some of the very few main impellers commonly used one I have given here. So what are those? Disc turbine, concave blade turbines, hydrofoil impellers, marine propeller. Okay. The number of impellers in a vessel mainly depends upon the container and also the volume. Usually 2 to 3 impellers may be connected to a single shaft at a difference of about 1.2 times the impeller diameter. Okay. Based on impeller diameter, they are going to fix. Then disc turbines. Okay, this particular disc turbines and the concave blade turbines are meant for gas dispersion for bulk mixing. Okay, whereas hydro foil impellers with the larger blades, about 50% of the vessel diameter, are more effective in that particular case. And further, vigorous agitation. Here, what happens here? Vigorous exhibits a shearing effect and causes damage to shear sensitive cells. Okay. Then here, the mixing contents with any fermenter is complex and influenced by several factors such as medium, density, size, and geometry of the vessel. Hence, stir tank reactions are provided with agitators having multiple impellers to give well-mixed homogeneous environment condition. Okay, so I hope you're able to understand about these agitation or maybe agitator or about the impellers. As I told, here different varieties of the agitators you are going to get, and like this is the disc turbine. Okay, okay, this you can see here, this is the disc turbine. Okay, let us understand here. So this is the disc. One second. This is the disc turbine, and here you can make out this is the disc turbine, the upper view of the disc turbine, and this is the concave beaded turbine. The next one is the hydrofoil, and next one is the marine propeller. Okay, like this, so many other will be the disc turbine, concave beaded turbine, hydrofoil impeller, and the marine propeller. You can make out here the other views. Side view, this is the disc turbine, and this is a wand disc turbine, and this is the variable pitch open turbine, and these are the marine propeller. So you need to keep in your mind these 4 different types of impellers: disc turbine, concave blade turbines, hydrofoil impellers, and marine propeller. These are different impellers which is usually used in the fermenter vessel. And also this blade depends upon the what type of product and what type of organisms you're using, okay, in the fermenter vessel. So in general, you can write for this agitator or impellers for 2 marks. The agitator is required to achieve a number of mixing objectives like bulk fluid and gas phase mixing, air dispersion, then oxygen transfer, heat transfer, suspension of solid particles, and maintaining uniform environment throughout the vessel contents. For all these purpose, we are using here the agitator or maybe a impellers. The process is known as agitation. All right. The agitators may be classified as disc turbines, vane disc, open turbines, a variable pitch, and marine propellers. Or you can write in short propellers. You can write this one for 2 marks. You can same information. You can elaborate for 5 marks also. I hope you understand. Right? Okay, fine. This is all about agitator. Next, let us understand about the bioreactor baffles. Okay, this is the baffles. Okay, vertical projection to the inner side of the fermenter vessel. Okay, so you can make out here, for example, ಒಂದು ಬಕೆಟ್ ಅಲ್ಲಿ ನೀರ್ ತಗೊಂಡು ಡಿಟರ್ಜೆಂಟ್ ಎಲ್ಲ ಹಾಕಿದಾಗ ಮಾಡಿದಾಗ ನಾವು ಆ ಬಕೆಟ್ ಅಲ್ಲಿ ಇರುವಂತ ನೀರನ್ನ ಚೆನ್ನಾಗಿ ಮಿಕ್ಸ್ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ಮಧ್ಯದಲ್ಲಿ ಸುಳಿ ತರ ಬರುತ್ತೆ. ಆ ಸುಳಿ ಏನಂತೀವಿ, ಸ್ವರ್ಲಿಂಗ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಆ ಸುಳಿ ಅಂತ ಬಂದಾಗ ಅಥವಾ ವರ್ಟೆಕ್ಸಿಂಗ್ ಆದಾಗ ಏನಾಗುತ್ತೆ, that will damage the cells and which hinders the product formation. For that reason, we are using the baffles. Baffles ಇದ್ದಾಗ ಏನಾಗುತ್ತೆ, ಆ ಸ್ವರ್ಲಿಂಗ್ ಆಗೋದಿಲ್ಲ, ಆಮೇಲೆ ಸುಳಿ ತರ ಸೆಂಟ್ರಲ್ಲೇ ಒಂದು ಆಗೋದಿಲ್ಲ. Okay, for that reason, we are using here the baffles or maybe a bioreactor. When baffles are kept over there uniformly, the mixing uniform rotation of the liquid can also be observed. So you can make out this the agitator may be a shaft, and baffles will be put in this particular level. Okay, the baffles obstruct the flow in the vessel to help mix the contents properly. Okay, now let us understand about the baffles theoretical part. Fermenter vessels are generally provided with 4 equidistantly placed baffles. You can make out here 4 distantly that will be placed. 1, 2, 3, and the 4. Another one will be here. So the baffles will be placed to the interior of the fermenter vessel. The fermenter vessels are generally provided with 4 equidistantly placed baffles. These are plates projecting into the inner space of the fermenter vessel from the inner wall of the vessel. All right. These are meant to prevent swirling and vortexing of the fluid. This is key word you need to underline and to keep in your mind. Vortexing may cause cell damage in many cases where the cells are fragile and without thick cell walls means that time automatically what happens? The cells will die. Okay, for that reason, to prevent all those things, we are used here the baffles. The baffle width may be 1/10 or maybe 1/12 of the diameter size. And these are used to break the vortex formation, okay, in the vessel, which is usually highly undesirable. It changes the center of gravity of the system and consumes additional power. Okay, I hope you're able to write about the baffles for 2 marks. I hope you understood what's the purpose of using the baffles or what's the purpose of fitting these baffles inside the fermenter vessel. As I told, if I'm going very quick, please pause this video, stop this video, then go through the content. So for the example, in short, we need to write about baffles means you can write 4 baffles are normally incorporated into the agitated vessels of all sides to prevent a vortex and to improve aeration efficiently. Okay. These baffles are metal strips roughly 1/10 of the vessel diameter attached radially to the inner wall. The agitation effect is only slightly increased with wider baffles, but drops sharply with the narrower baffle. So this is all about the baffles. I hope you're able to write about the baffles in the examination for 2 marks or maybe for 5 marks, right? Now let us understand about the anti-foaming agent. Up to here, you all have in your syllabus that is about the baffles, impellers, and the sparger. Now let us also understand about the anti-foaming agent. As this is our stir tank fermenter, we are able to observe the impellers, the baffles, and the spargers. There will be chances of the foam formation chances usually happens. Now let us focus on what is this particular anti-foaming agents. During fermentation process, foaming is the common problem that is due to proteins in the medium, right? In the nutrient medium or nutrient component medium are byproducts by the microorganisms. Automatically, there'll be a foam chances will be there because there is a aeration is there, agitation is there. Due to all those conditions, automatically foam formation will occur. If foaming is not controlled, that affects the product yield and also block the air filters, and due to the loss of aseptic condition, fermenter become contaminated. For all these reasons, we need to use the anti-foaming agents. Once the foam occurs, immediately anti-foam must be treated in the fermenter vessel. All these are automated, okay? If no foam is not controlled, what happens here? If the foaming is not controlled, that affects the product yield. Who wants to lose the product, right? So definitely they'll go for anti-foaming agents, inlet pipe. And that will also block the air filters, right? And due to the loss of aseptic condition, fermenter become contaminated. For this reason, we need to add the anti-foaming agents. So controlling methods like modification of the media components is also very important. The media or what are the protein content or carbon nitrogen sources, what we are providing in the form of the nutrients chemical, that should not give too much foam in the fermenter vessel. If they give too much foam in the vessel, then that hinders the product formation. We are unable to get the good result. A desired product is not possible. So we need to modify the media components in such a way that that should give a less or minimum amount of the foam. Then use of mechanical foam breakers can also be used in a fermenter vessel. Then addition of the chemical antifoams. Then adjusting the different parametric such as proper temperature and the pH if adjusted means that time also we can avoid the foam formation. Then chemical antifoams are like surface active agents that reduce the surface tension that binds the foam together. Okay, some of the chemical antifoams can be used. And some of the natural antifoams like plant oils, soya, sunflower, deodorized fish oil, minerals, Okay, oil, mineral oil, etc. can also be used. Some other synthetic antifoams are like silicones, polyalcohols, then alkylated glycerols, esters, fatty acids, or fats, or sulfonates. All these can be used as antifoaming agents. I hope I made this about antifoaming agents or antifoam very clear. In the examination, if they ask antifoams, compulsory you must write. Now let us focus on other parameters in the particular fermenter vessel, like inhibitors. To get specific products, we can use this particular inhibitor. These are used to redirect metabolism towards the target product and to decrease the fermentation of other metabolic intermediates. So in the fermenter vessel, we are adding this inhibitor to decrease the other metabolic intermediates. We are using this particular inhibitors, which focus mainly on our target products. Then coming to the oxygen. Depending upon the oxygen required by the organism, pure air must be supplied whenever there is a demand of the oxygen. Okay, that is very, very important. Oxygen concentration must be also monitored. And also sometimes in the fermenter vessel, we can also use the chelators. These chelators are the chemicals used to avoid precipitation of the metal ions. Okay, for example, we can use here EDTA, citric acid, polyphosphates, which are used in the low concentration as chelators in the fermenter vessel. And next one is inducers. Even sometimes you can use in the fermenter vessel inducers like starch for amylase. Here, why are we using here inducers? Majority of the enzymes used in the industrial fermentation are inducible and are synthesized in response to of inducer. When you add the inducers, there what happens? The product yield will be more and also very specific. That's why we are giving the inducers in the fermenter vessel. For example, if you want amylase means you need to provide the starch. Starch which is going to act as the inducers. So to obtain the pectinase, you need to put the pectin so that you'll get the good yield of the pectinase. And simultaneously for cellulose, you need to use cellulose. Okay. Even you need to keep in your mind some of the precursors can also be used in a fermenter vessel. It means these precursors which are directly incorporated into the desired products to enhance the yield of the product. If you want to enhance the yield of the product, you can go for using the precursors. And these precursors like starting compounds, okay, igniting compounds for the production of the product. So that is these precursors are the starting compounds for the synthesis of the final products. For example, if you want penicillin G means, you can put precursor as phenylacetic acid. You'll get very good yield of the penicillin G. Like that, if you want streptomycin, go for adding the proline as a precursors. If you want good yield of the vitamin B12, put cobalt in optimal concentration, you are able to get that particular product of the vitamin B12. Corn strip liquor can also be used for the production of the several types of penicillin, but addition of phenylacetic acid leads to the fermentation of the penicillin G. Okay, corn strip liquor also which is giving a good yield, but if you add the phenylacetic acid specifically as a precursor, we are going to get the very good yield of the penicillin G. I hope you understood about this particular precursors here. And also you can use some of the growth factors. Some of microorganisms cannot synthesize the required cell components themselves and need to be supplemented. Okay, that time we can go for the addition of the growth factors in the culture medium or a vessel like thymine, biotin, amino acids, plant or animal extract in the pure form. All these we can put as a growth factor to obtain a good product. And the next most important one is the buffering agent, the regulation of pH. Okay, the regulation of pH is also a very important factor in order to achieve optimal productivity, right? Control of pH by buffers, very, very important. If the pH is not controlled properly, you're not able to get the product. Optimum pH, which gives the which allows the optimal growth of the microorganisms and also which gives the good yield of the product. Before subjecting medium for the sterilization, it is necessary to check the pH of the medium. And to make it desired pH, by adding acid or alkali. Okay, that is very, very important. Before also and during the process also, one has to check one has to adjust the pH. To adjust the pH, nowadays almost all the bioreactors are automated one. Acid and base reservoirs will be there. Automatically pH will be checked. Before pouring to the vessel also, one has to check the medium pH. Many media adjusted to 7. pH 7 will be maintained, okay, by the addition of the calcium carbonate or maybe a phosphates. Also, through balanced use of carbon and nitrogen source will also form the basis for pH control. So this is about the pH regulation or maybe a buffering agent. Further, frequently the pH of the production medium changes in 24 to 48 hours. Why this changes? Due to the certain metabolites released from the growing cells. When we take the growth of the cells or maybe the particular graph, lag phase will be there, log phase will be there, and death phase will be there. In there, you can see the automatically so many changes can also be observed. Like that, even pH during the period of time also, pH also will vary. One has to adjust. It is necessary to maintain the optimal pH of the broth at continuously. The optimal pH must be maintained. This can be achieved by adding alkali or acid after testing a sample at different intervals, and that can be buffered by a calcium carbonate or addition of NH3 or NaOH or maybe H2SO4. In the modern automated system, the pH is monitored by pH probe. In the fermenter vessel, at the top of the lid, there will be the presence of the pH probe for the acid and the base, which is reloaded periodically, and corrective measures taken by the systems only. As most of the bioreactors are automated, so acid base will be controlled there only. Now let us focus on the temperature regulation. Okay, which is very, very important, because how the pH also will vary, like that even temperature also will vary due to so many reactions will be happening in the fermenter vessel. So temperature has to be maintained at the optimal level throughout the bioprocess, and it's the most important parameter to monitor and control in any process. This is very, very important. Otherwise, what happens? The temperature regulation is not happened properly. What will happen there? Automatically, the product will be reduced. Our organisms are unable to grow properly. Organisms are unable to give the product. Organisms are unable to react. Optimal temperature is maintained, then only they are going to be able to utilize the nutrients and also give the products. Okay, and there'll be a jackets will be there, right? Jacket which provides the annular area for circulation of the constant temperature of water, which keeps the temperature of the bioreactor at a constant level. Cooling jacket and heating jacket will be provided in the fermenter vessel, and the heat is generated during the fermentation, that is due to metabolic activity of the microorganisms. Sometimes the temperature will be generated in the fermenter vessel due to the metabolic activity, and also due to the mechanical agitation process, the temperature may be produced. And temperature in the fermenter is measured by mercury in the glass thermometer, a biometallic in the thermometer, a pressure bulb thermometer. Like that, there are different parameters of thermometers will be there. During metabolism, in a closed system, microorganisms release considerable degree of the heat, and hence the fermenter needs to be cooled. Okay, this is achieved either by spraying cold water through the outlet jacket of the vessel. Okay, in addition, in rare cases, it may be necessary to use internal coils or tubes through which cold water is passed to remove heat. Okay, to reduce the temperature, we are going to send the cold water. Okay, cooled water will be sent, and to increase the temperature, several heating coil device will be used to give the temperature. Okay, thermophilic organisms may require high temperature for the growth. In such cases, it may be necessary to pass steam to raise the temperature using internal coils. Okay, steam valve will be there, steam inlet pipes will be there. Through that also we can send the steam. Okay, so this is all about the temperature regulation. So in this class so far, we went through about the sparger and about the impellers and about the baffles, and also about the temperature regulation, right? And about the pH regulation, inducers, precursors, right? Growth factors, so many things we went through. I hope this class is very useful. And if I've gone very quickly, please pause this video and go through this one, okay? Today I have covered related with the impellers, baffles, and the spargers. In the next class, let us understand more detail about the different types of the bioreactor. Okay, so today we went through about all these, that is aeration, agitation, temperature regulation, foam control, pH regulation, right? I hope you understood the entire concept. During your free time, please go through all these content and also acquire more knowledge. I hope I made this particular aeration, agitation, and about the baffles crystal clear. If any kind of doubts, you can reach to me and you can clarify your doubts. Okay. All these you need to keep in your mind for the examination purpose and start studying. Okay. Can make a for notes also. That will be very useful. Okay. I hope I made your day with the knowledgeable fermenter information. Thank you everyone for watching this video. Thank you.